Okay.
Mm, Hello, my name is Maria, and today I'm going to talk about my research project called Ancestry-conditioned generative models for genotyping. First, I'm going to do an introduction, and I'm going to explain the dataset used. Then, I'm going to talk about deep generative models used, and finally, I'm going to draw some conclusions. I will start explaining what is population genetics and some important background concepts in order to understand the work done. Population genetics is the science that compares the existing differences between genomic sequences of humans from distinct populations. We refer to population groups as ancestries. Genomic sequences are made up of DNA, and around 2% of positions in the human DNA sequences vary between individuals, and these are named SNPs. So the SNPs are the positions of the DNA where there can be a variation. And these positions can be codified in a binary system with zeros denoting the common variant and one the minority variant. For example, in the image, we can see three different individuals where each one has their own DNA sequence. And in the first position, we can see a SNP, which is a change of the letter in the specific position. The frequency distribution of those SNPs and the correlations between them are linked to different population groups, such as Europeans, Asians, Africans, etc. This is caused by the facts of having different founders, migrations, and genetic variances along with history. So that the different ancestries can be differentiated based on their SNPs. There are two main challenges when dealing with biology, medical, and genomics data. The first challenge is the underrepresentation of several groups of populations. So currently, the majority of the human individuals included in genetics studies are Europeans. And this is a critical problem because this introduces huge biases in the results. Thus, some medical treatments work well for Europeans, but not for their populations as Asians or Africans. So, to sum up, some population groups do not have enough data to be included in studies. And the second challenge is related to data privacy that hugely limits availability of datasets because it contains sensitive data. So generative models can be useful in such scenarios because they can mitigate the lack of data sets for specific population groups. And furthermore, generative models can be easily shared once trained. Now I'm going to explain the dataset that I use to train the models. The dataset is based on human labeled genomic sequences that contain SNPs. The SNPs are codified with zeros and ones. A zero indicates the common letter and a one a variation. Each sequence belongs to a specific individual, and the labels are the ancestries of those individuals. And in this work, I use four different ancestries, in particular Europeans, Africans, East Asians, and South Asians. And the dimension of the dataset is 3,366 individuals and 5,000 SNPs. And here we can see a simulation of the dataset where each individual has 5,000 SNPs and is from a particular ancestry. The most common plot used to analyze data of different population groups is the principal component analysis. This technique makes clusters according to population. And in this image, we can see the PCA of the original data with four clusters, which are the population groups. And we can see that there is some noise that are the layered samples that are far from the clusters. So as a first step, we decided to remove those samples, and the resulting PCA is this one. I also try different dimensionality reduction techniques like linear discriminant analysis, neighborhood component analysis, and UMAP. The three methods are also dividing the data into clusters. Regarding the evaluation metrics, I use the PCA, where I project the generated data on top of the real data. And the generated data should fit inside the cluster of a particular ancestry. I also use the mean square error and binary cross entropy between the means of the real and generated SNPs. And finally, I use these three classifiers that aims to classify between real and generated samples. And I use the accuracy of those as an evaluation metric. It's important to mention that we want a low accuracy because we don't want the classifier to know when the samples are real or generated. And a low accuracy means that the generated data is very similar to real one. That is our intent. And I trained every model 5 times with 5 different seeds, and I computed the mean and standard deviations of the accuracies to have more accurate results. And now I'm going to explain the different generative models used to generate synthetic data. The first one is a generative adversarial model that it's called GAN, and it contains two networks, a generator that generates synthetic samples, and a discriminator that classifies between real and fake samples. And both networks are simultaneously trained. The first model that I implemented is a GAN per ancestry. So if we have 4 ancestries, 4 GANs are trained. And in this diagram, we can see the scheme of a particular GAN. The input of the generator is random noise, and the output is the generated data. And the input of the discriminator is the generated data or the real data from a particular ancestry. And the output is a label 1 or 0, indicating if the sample is real or fake. The loss of the generator is a binary cross entropy between the predictions of the discriminator when the input of the discriminator is the generated data with a level of real because the generator wants to cheat on the discriminator. And this next loss will be explained in the next slide. And the discriminator is computed doing the mean of 2 binary cross entropies. One with the predictions of the discriminator when the input of the discriminator is generated data with the level of fake, and the other with the predictions of the discriminator when the input is real data with the level of real, because we want the discriminator to distinguish between them. And another loss added in the generator is a loss that, well, I call it a mean matching loss, but it's based on the maximum mean discrepancy that I will explain it later. And this mean matching loss consists of computing the mean of the SNPs of the real samples and generated samples, and then computing the mean square error between them. Then the generator loss is computed with the mean matching loss plus the binary cross entropy. The second model implemented is a conditional GAN. There are 3 main differences with the GAN. The first one is that the conditional GAN is trained with the data of all the ancestries, although each batch contains data of just an ancestry. So we are training just 1 GAN. And the second difference is that the ancestries are concatenated with one hot encoding in the input of the generator and discriminator. And the third difference is that the output of the discriminator is fake if the input sample is generated, or the ancestry if the input sample is real. And as the output of the discriminator is not binary, the loss now is a categorical cross entropy. Regarding the results, the only one that separated the generated data into clusters is the GAN per ancestry with binary cross entropy plus the mean square error. And in the plot, we can see the PCA of the generated samples projected into the real ones, and the dots are the real samples, and the cross is the generated samples. And we can see that the generated data is separated into clusters, but the samples are not all inside the real clusters. And also the accuracies of the classifiers are almost 100%. So although the samples are separated into clusters, the results are not good. The third model, the third deep generative model is a generative moment matching network that is trained by minimizing the maximum mean discrepancy, which is a frequentist estimator that tries to determine if two samples come from the same distribution. And the aim of the model is to match all the orders of statistics between samples from the model and the input dataset. The difference with the GAN is that there is no discriminator. We just have one network that generates synthetic samples. And this is an advantage because the training is easier. We use one network per ancestry as before. So we train 4 times the network, one per ancestry. And in the diagram, we can see the scheme of this model. The loss is computed with the mean of the generated samples and the mean of the real samples from a particular ancestry. I try 3 different losses between the means, the mean square error, the binary cross entropy, and child divergence. And I also try adding the covariance. So I computed the covariance of the real and fake samples, and then the mean square error between them. And an important thing to mention is that the mean of the real samples are pre-computed for each ancestry to save computing time. And then I also try a conditional generative moment matching network. The difference is that now we use the data of all the ancestries, and the same as in conditional GAN, the labels are concatenated with one hot encoding in the input of the generator. And the losses are the same as in the moment matching network. The results of the generative moment matching network per ancestry are very good. First, if we compare the results of just using the mean, the best one is with the mean square error because the errors are the lowest one and the accuracies too. So the next results are computed with the mean square error. And then if we compare the mean plus covariance using the mean square error, the results are very similar and the PCAs too. The PCAs are very good because the generated samples are fitted inside the real samples. But adding the covariance means more parameters and more training time. So as the results are very similar, we would say that the best one is without using the covariance. The results of the conditional moment matching network are not as good as the previous ones. The binary cross entropy and child divergence did not form clusters, and the mean square error between the means and with adding the covariance did form clusters, but the generated data is not perfectly fitted in the real data. And also the accuracies were almost 100%. And the motivation of privacy from generative moment matching network is that you can publish the train model, so it's possible to generate data without needing the real data. But you can just generate new data in the train domain. So a solution for that is to compute specific features from the data and publish the features and the model so that it's possible to train the model. And for that purpose, I decided to use random features. That is what I am going to explain now. So I compute random features with a linear layer and a cosine or a ReLU. And the network is not trained in order to be random. And the basic idea is that when the number of features tend to infinite, the features approximate the same space as some kernels. So I try different output sizes, in particular multiplying the input by 1, by 5, by 10, and by 12. I tried the random features with the generative moment matching network. As we can see in the diagrams, the data is only used to compute the features. And in this model, we don't even need the features. We just need the mean of the features that it's called sketch. So, for example, a company that has the real data could compute the features, and in this approach, just send the mean of the features. That is what is needed to train the generative moment matching network. The only method that provides good results is the generative moment matching network per ancestry with random features. And first of all, I would like to mention that the random features network was working better when doing a restart of the wave every 500 epochs. So that means that the real data is needed to compute the features again, or a schedule already computed. So to understand the motivation of that, I will explain two examples. The first one is that, for example, a hospital gives the data every 2 months. So every time that the hospital releases the new data, the features are recomputed. And the second example is, for instance, that several hospitals are giving the data, so the features need to be recomputed for each hospital data. And as I mentioned, the network is composed by a linear layer or a ReLU, a linear layer and a ReLU or a cosine. And comparing the results in the first table, we can see that the ReLU gives better results as the accuracies are lower and the errors too. I also try different output dimensions using the ReLU as activation, and we cannot see a clear difference when multiplying the input by 5, 10, or 12. The quantitative metrics are similar, and the PCAs too. The PCAs are very good as the generated data is fitted in the real data. So we would say that the best one is multiplying by 5 because it has less parameters and is faster. Here we can see the architecture of the generative moment matching network per ancestry using random features. And as mentioned, the input of the generator is random noise. And the generator is based on a linear layer, a ReLU, a batch normalization, a hidden layer, and a binary quantizer that binarizes the output samples. Then the features are computed from the data with a network, and finally, the loss is computed with the means. Finally, as a conclusion, I would say that the generative moment matching networks are working better than GANs, and that working per ancestry works better than conditionals. Generative moment matching network with and without features work similar, but features have an important advantage that preserve data privacy. And in these four images, we can see the PCAs of the generative moment matching network trained for each ancestry and with random features. And we can see that the generated data is very similar to the real one. So that the generated data has a very good quality. And as a future work would be to improve random features methods, for example, training the features or using new methods as kernels. And also to see why the conditional models are not working. And as I showed you very few results, now I'm going to show you a short video of some more. and thanks for your attention.
Thank you very much, Maria. Let's move to the questions. Javi, do you want to start?
Yes, wait a second that I put my volume down. Okay, can you hear me?
Yes.
Okay, Maria. So congratulations for your work. I
Ferran,
also found it very interesting. So...
Stop recording.
Stop the recording, Ferran. Thank you, Jordi. Okay, so I have some